All right, welcome everyone to this week's Autonomy Talks. This week is a great pleasure to have with us Ritu Raman, who is the Eugene Bell Career Development Assistant Professor of Mechanical Engineering here at MIT. Something about Ritu. Ritu received her Bachelor of Science from Cornell and a PhD from the University of Illinois, Urbana-Champaign. She then spent a while as a postdoc at MIT, working with Robert Langer. So today we have a very exciting topic. Ritu's lab is centered around 4D tissue engineering of biological actuators, and applications span medicines and machines in general. As you've seen from the bio I've shared, Ritu has won many awards, actually too many to list them, but as a junior faculty, I'm very impressed by all these awards. And also a nice thing is that she has a cool MIT Press book on biofabrication. You should check it out. In general, Ritu is passionate about increasing diversity in STEM, and she has led many initiatives, and I invite you to follow her on her social media. She's quite an influencer for us academics, I would say. Today, she's going to talk about leveraging biological actuators for soft robotics. This is a very exciting topic, and I'm eager to hear more about it. That's why I give you the stage. Ritu, go ahead. The stage is yours.
Great. Well, thank you so much for the kind introduction, and it's great to be here today. Joel mentioned that the norm for the seminar series is if you have a question, like a clarifying question or anything in the middle, feel free to just raise your hand or interrupt in case I don't see it. Just because since it is a very sort of multidisciplinary topic, there might be some concepts I mentioned, particularly those in biology, that are not clear or not resonating. So I would rather you clarify them when you're confused rather than waiting till the end of the talk. So that's the rule set or the disclaimer. So as Joel mentioned, I'm a mechanical engineer by training, and I'm in a mechanical engineering department. And my broader goal as an engineer is to think about whether we can add biological materials to the set of materials we think about building machines and other functional things in our world with. And the reason to do this, right, is that biological materials can sense and respond to their surroundings in real time. And so they have this dynamic adaptation that is really beautiful. And in particular, the class of biological materials that we think about building with are actuators. So specifically thinking about how pretty much every animal, whether it's swimming or walking or flying, is doing that powered by skeletal muscle. And that muscle is controlled by upstream motor neurons and also has embedded sensory neurons that help us accomplish closed loop feedback control mechanisms, like picking up something and putting it down and knowing how heavy it is. So that sort of behavior is really beautiful and what we want to try to mimic. And if we could build those sort of motor control systems from scratch in a petri dish type of environment, it would not only be interesting as something that we could apply to robotics, but it would also help us understand a little bit more healthy human mobility, how to restore it after disease or injury. So that's what we tried to do in my lab. We have sort of three big areas that are spanning what we call 4D tissue engineering, in other words, just building tissues that are composed of multiple cell types. And we develop tools for fabricating those types of tissues. We apply those for applications in robotics, and we also apply them to understand more about medicine. So in this talk today, I'll touch a little bit on all of these areas, but since the specific focus of this group is on the robotic side of things, I'm going to start there and see how far we get. And then with the remaining time, I'll kind of walk through what we have remaining. So when we're thinking about muscle as an actuator, I think it can be really helpful to first put it in the scale of other things that we know, right? If we plot the force that a single unit of an actuator can produce by its size, log plot, we see that most of the things we're used to building with operate at the millimeter to centimeter scale, right? They produce reasonably large forces, some more than others, but they're all operating sort of in this regime. And the really important thing is that for muscle, even though the muscles in our bodies are quite big and they are operating in this size regime, they're composed of several individual cells. Each of those cells is an independent actuator, and each individual cell is on the micron scale. So that means you have tremendous scalability in terms of how far down you can go and get efficient actuation at much smaller length regimes. And you can also stack a bunch of muscle cells together and get something that performs really well and comparable to traditional other types of actuators we have. But it's actually better than a lot of other actuator systems we have in some regards, namely the size and energy density of the energy source that it uses. So muscle is powered by sugar. Sugar is a very dense biodegradable energy source. If you compare that to, say, like a pneumatic actuator, where the actuator itself is not very large, but the compressed air gas tank next to it is quite significant, when you account for the power to weight ratio that accounts for the size of the battery, muscle actually outperforms nearly all traditional actuators in that regard. And then the other thing, of course, is that it can always sense and respond to its environment. So it can heal from damage, it can exercise and get stronger. And these capabilities, right, for the most part, are not true of other types of actuators. So this is where biological actuators are really excelling, right? They give us scalability, they give us efficiency, and they give us adaptability. And so that's why we want to build with them. And when I started kind of working in this field, I was very lucky to have inspiration from a range of papers from Kip Parker's lab at Harvard, where they showed that if you grow a sheet of cardiac muscle cells, so those are the muscle cells that are in our hearts, on a thin polymer film, all of those muscle cells, when they beat, can deform that film, and you can get the sort of propulsive locomotion of that film through a liquid. And what they've realized over the years is that the closer you get to mimicking the biological architecture of a real swimming animal, so, getting the fins the right shape, getting an agonist and an antagonist muscle, the closer you can do that, the better you are to achieving the swimming speeds and efficiencies of real animals. So this work is really beautiful, really exciting, but the kind of big takeaway for me was thinking about the fact that cardiac muscle, while very useful for this sort of application, is not designed to be turned off, right? It's in our hearts, which means it's constantly on, it's constantly beating. And the only control you have over it is how fast it's beating. Our hearts beat faster when we're running versus when we're walking, right? But you don't want it to turn off because that would be very bad. But if you are thinking about a robot, that's not, you don't want something that you can control only its speed, but not whether it's actually doing something, right? So when we think about all of the voluntary motion we have in our lab, in our lives rather, if I want to pick up this glass, I want to hold it for some period of time, and then I want to put it back down, then I need to be able to have on-off control of different locations of muscle at different times. And so for that, we don't want cardiac muscle, we want skeletal muscle. Which is what we use all throughout our bodies, our arms, our legs, our postural muscles to control voluntary movement. So skeletal muscle is a little bit different in architecture from cardiac muscle. Rather than being a sheet of cells that beats at the same time, each cell is a really long cell or fiber. It looks like, a thin hair, but much smaller. And the hair-like cell has two proteins, actin and myosin, that normally look like this. And then when you send an electrical input from an upstream nerve to the muscle, they move closer to each other. So they shorten. And so this long cell gets a little bit shorter. And if you stack many of these cells in parallel, like what's shown here, you can get a really large reproducible force that's produced. And then how do you control those cells? I mentioned that each muscle cell has one neuron that's connected to it that tells it to turn on or off. And then that one neuron might control a few muscle fibers at a time. In our hands, that is usually a fairly small number. So one neuron might control, 10 or 15 muscle cells. That's what we call a motor unit. It's a pretty small motor unit because you want really fine motor control in your hands. And then in your legs, one neuron could control hundreds or thousands of muscle fibers, because there you're just trying to recruit large populations of muscle at the same time to do things like walking and running, but you're not as worried about fine motor control. So that each motor unit is an independent actuator. And then in each muscle, like your bicep or your tricep, you usually have multiple motor units. So there's a lot of redundancy in the system. If you hurt one part of your bicep, you're still able to move your arm because the other motor units are still functioning. So that's a sort of high-level biological overview of the kind of system that we have. And I see something pop up in the chat here about controlling motor actions. Yes, so in the brain, you have, so this right here are your lower motor neurons that are in your spinal cord. And then these are connected to upper motor neurons in the motor cortex of your brain that are related to motion planning and execution. So I'm mostly going to focus the talk today on stuff peripheral, so just on off, and not about like decision making or planning, which is out of the scope of the kind of work that we do. But great question. Okay. All right, so we want to make that kind of architecture, and we ask ourselves, how might I build something like that in the lab? And we say, okay, well, if I wanted to build muscle, the way I would do it is by starting off with a bunch of muscle cells. We use both mouse and human cells in the lab. We mix together those muscle cells in essentially a soft protein jello, which has a bunch of proteins that mimic the native extracellular matrix environment of the muscle cells. The cells proliferate. So the cells are shown in blue here. These little blue dots are all individual cells. They proliferate and then they fuse together to form these long muscle fibers. So this long green line here is now a single mature muscle fiber. And then you have many of these fibers that are stacked together, right? And they're pointing in the same direction so that when they contract, they can all contract at the same time. And the way we get them to point all in the same direction is by imposing forces on them. So we stretch them around some sort of skeleton. That skeleton imposes a passive tension force on the muscle. And so all of the fibers align along that axis of tension because most cells in our body are responsive to mechanical forces in their environment. Now, what we need to do then is to control the contraction of the muscle. And in the absence of neurons, how do you do this? Well, you say, well, neurons are really opening and closing a voltage-gated ion channel in the muscle membrane. So they're opening and closing an ion channel that lets in calcium ions. That calcium ion channel change causes the muscle to contract. So if I don't have neurons, the way to do it, which in the first generation of robots, we didn't have the neurons, the way to do this is then to genetically modify the muscle cells to have a light-gated ion channel instead, so that when you flash a light on the muscle, you can get it to contract, and then you can get this sort of large motion where when every time the muscle contracts, you can see that the skeleton deforms or like does something interesting. So once we had this sort of basic functional unit worked out, over the years, we started developing a range of walking robots, first one-legged, then two-legged, and then some with like wireless electronic control, where we essentially showed that if you shine light on a muscle, you can control the direction in which the robot walks, and you can get it to move and turn and rotate in 2D space. And while we focused primarily on walking, other folks have used the protocols that we developed to turn the linear actuation of skeletal muscle into swimming robots and pumping robots and gripping robots. The actual robot itself doesn't matter, right? The baseline technology is the same, which is, can I get a muscle to contract and do something useful? So this is great, but one sort of common design limitation that all of these muscles had is that essentially, you know, they were kind of one-off designs, right? You would make your muscle and you would say, okay, I think I want to make a pumping robot or something. And you would say, okay, all right, I want, you know, a tube. It should be approximately this stiff, and the muscle will make it deform a little bit, but nothing was necessarily like optimized about any of these designs. It was just a very early stage of the field. So it was enough to say, isn't this cool? We can get muscle to do X or Y. But those of us who are, you know, working maybe in more mature principles or mature fields of robotics know that that's stops being acceptable after a certain point, right? After you've done all of your proof of concept work, now you need to think about, how do I get my muscle-powered robot to do the same thing every time? And how do I maximize the stroke output of the muscle and convert that into some useful function of a robot? And once I do that, I can say, right, that I truly understand how to design a muscle-powered machine. I'm not just making a one-off thing every time. So to do this, we first made a muscle tissue, and then we took it off of a skeleton and just had it free floating in media to see, like, how does it move and what is the maximum motion that it can generate? Because this is theoretically the most the muscle can move, right? It can't move much more than this. So if we do this, what we realize is that the muscle is mostly contracting in this direction, which makes sense because most of the fibers are aligned along this long axis. So they're mostly contracting in this direction. And any other contraction that you have is essentially wasted motion. So if you have x-axis contraction, that's great, and that's what you want. But if you convert that contraction into displacement that's like in the y or z direction, or rotation about x, y, or z direction, that's not actually doing any useful work for you. And so we said, okay, so when we think about how to design a skeleton for this muscle, what we want to do is build a skeleton that has minimal resistance to contraction in this x direction, right? So very compliant in this x direction and extremely stiff in all other directions. And if I did that, if I essentially model that as a spring, I could, and I have my my muscle is usually like a rubber band or ring. So I have like this part of the ring and that part of the ring. I can model the muscle flexure interaction by saying, if this is the stiffness of my muscle curve and this is the stiffness of my flexure curve, when I have a very compliant flexure, I will get a lot of movement, but not much applied force. And when I have a very stiff flexure, I will get not very much movement, but a large applied force. Okay. And so in our first sort of version of this, we decided to focus on this high stroke regime, and we designed a skeleton that's basically just a linear elastic spring. It's a big, floppy, extremely, extremely compliant spring. I think in this first paper, we were targeting something around 0.3 newtons per meter, so really tiny. And what we did then is said, okay, when the muscle contracts, because this is so compliant, it should essentially contract as much as it does when it's free floating, because you're so close to this regime here. And what we saw is, in fact, that this was true. So what you do is by putting it on this flexure, you make sure that the only motion that you're seeing, you're not getting all of this random motion in all different directions. The only motion you're seeing is in this direction that's parallel to the muscle fibers, and you don't lose rotation anywhere else. And actually the strain output that you get is much more reproducible. So let's say if they're floating around, you get a strain output of anywhere between 1 and 5%, depending on how your muscle is oriented. Here you get a contractile strain output of 6% every time, which is not incredible, but it's still pretty good, right? And on the order of like shape memory alloys and other things. Natural muscle has a strain output of closer to 20%. And I'll talk to you a little bit more about how we're pushing in that direction in the coming months. But essentially, we've done this and we've shown that you can get these really reproducible force outputs, which means that in our previous versions of our bots, when we had them on these little like cantilever type of devices, if the muscle scooched down even a few microns here or moved up a few microns here, you could dramatically change your readout of displacement. And so you'd get these really highly variable readouts. And so if you were trying to compare, say, diseased muscle to healthy muscle, or trying like a new growth factor or a new way of training the muscle, it would be very hard to tell the difference between what's a good muscle and what's a bad muscle on this type of platform that is so subject to exactly where the muscle is placed on the beam and also the contact area between the muscle and the beam. But if you put them on our flexure devices, because they're just a rigid post connected to a really compliant spring, you get very, very precise readouts every time and a almost 10x magnification in how much they move. And so because of this, you can very clearly distinguish between what's a good muscle and what's a bad muscle. And when you have a good muscle, you get the exact same displacement out every time. Okay, so this is really useful for robotic design and helps us then get really accurate measurements, not only of the displacement, but then we can also readily convert that into a measure of force, right? Because if we know the displacement and we know the spring constant of the flexure and the spring constant and the stiffness of the muscle, we can calculate how much force the muscle is producing over time. And we can also look at its frequency dependent response. So we see that if we stimulate the muscle at 1 hertz, it contracts and then it has time to fully relax before it can contract again. And then if it contracts at higher frequency, then you contract, and then by the time you can fully relax, you have to contract again. And so your dynamic range of motion is reduced, but your power output actually goes up. And the other thing we've kind of learned is that natural muscle has a sort of characteristic response curve where you turn on the light, it takes a couple milliseconds to get to its peak force. and then it stays at its peak force for some period of time, and then it relaxes down. And so in the past, it was thought that if you wanted to spend more of your time at this peak force, the only way to do it would be to operate at really high frequencies. But what we found is that if you just keep your light on for longer, you can, by changing the duty cycle of your input, essentially, you can keep the muscle at its peak force for arbitrarily long periods of time and decouple sort of your range of motion from the time you're spending at peak force. So you can contract the muscle, keep it at its peak force, and then relax it back down. And you have this really, really precise control of not only the force that's being produced, but also the dynamics of that contraction. Okay, and then the last thing we've done to sort of characterize this force characteristic is to think about how we can use our flexors to monitor fatigue in our actuators, because also we want to think about what happens when we try to operate these continuously. So what we've seen is that if we operate something for about 30 minutes continuously, they produce essentially the same forces for that whole period of time. But if you increase the frequency to about 4 hertz, they start off looking really good, but then the force drops off fairly quickly within about a few minutes. And by 30 minutes, you're barely moving at all. And at first we thought that this was something that was just like an intrinsic property of the muscle that we couldn't change. But some new data in my lab is showing that by exercising the muscle at different frequencies, you can actually tune the fatigue response quite a bit. So you can change that fact that or rather you can train the muscle at high frequency and then get it to be fatigue resistant at high frequency, depending on exactly how you train it. So there's more to come that's more sort of biologically driven in this space, but that work is still in progress, so I can hopefully share it with you next time. All right,
Ritu,
and then
there's a quick question from Alice. I think it's good to ask it now. Alice asks, how long do these muscles last, and do they have any degradation over time?
Yeah, that's a great question. And honestly, I should probably add it to the slide deck because people ask this question every time and I never think. So I'm going to write down to myself right now, add lifetime.
That's great.
But essentially, so this is fatigue in a short period of time. The question is, how long do they last? Typically in my lab, we keep muscles on the order of several weeks. However, we have seen that we can keep muscles alive and active over hundreds of days in the past. And so it's just a question of the thing that sort of mediates how long the muscle lives, right, is that every day you have to give it food. It's just like a living cell. So you give it media, you put it in a liquid media that has sugar and amino acids and growth factors that it needs. Over time, the cells, because they don't know that they're outside of the body, right? They just think they're in a tissue. And so in our bodies, it's very normal for cells to want to remodel their extracellular matrix environment. So they're always secreting different enzymes to break down the matrix and then depositing new matrix and doing all of these things. And so what we found is that if we just grew our muscles normally, they would sort of tear and break apart within a couple of weeks because these muscles had secreted so many enzymes called proteases, so things that cut up proteins, that the matrix itself, the gel that the cells were in, would fall apart. But over the years, we realized that if we included a protease inhibitor in the media, so something that stops the activity of this enzyme, then the tissues could sort of be kept indefinitely for hundreds of days. However, we haven't quantified how the performance drops off over hundreds of days, but it definitely does. So that's something to think about and work around. But for the purposes of this talk, I would say very comfortably, I can say they work very well on the order of several weeks. All right, so hopefully that answers that question. And so the sort of design of the skeleton that I talked about gives us an idea of, okay, how do I maximize the range of motion that the muscle is producing? And then the next question is, how can I enhance the actuator efficiency even further, right? Because what I have right now is I'm making these muscles that are kind of like rubber bands, and then I'm stretching it around some rigid skeleton, and it's just like a friction fit, right? There's nothing to stop this muscle from sliding up and down or twisting this way. And so the transmission of force from this very soft actuator to this very rigid skeleton is not very reliable. And so our thought was, okay, well, in our bodies, how does this work? We actually have muscle that is covalently linked or bonded to bones via tendons, right? And so we have a muscle tissue, it's got a tendon at either end, and that tendon is sort of integrated with the bone. And so if we really wanted to enhance the efficiency of our actuators, we would want a bone, tendon, muscle, tendon, bone unit. And right now we just have bone, muscle, bone. So the question is, how do we make that tendon? So the first thing we did is think about the fact we looked around our neighborhood here at MIT, and luckily one of our next door neighbors is Zhuan Hezhao's lab in our MECE department, and his lab develops a lot of tough adhesive hydrogels for medical applications. Like, oh, if you had an injury and two tissues sort of tore or came apart, you have these tough adhesive hydrogels that can be used to stick the tissues together. And so they were using it for a lot of really cool medical applications, and we were thinking, that's great, but would it also be possible to take these tendons and stick them to some of the tissues that we're making, not native tissue, but engineered tissue in the lab? And if we could do that, essentially what we could do is take our muscle rings, cut them up, stick two little tendon pieces on either end, and then mount them onto our skeletons, right? So that's exactly what we did. We took our muscle tissue, which is shown in pink here. We have two tendon pieces that are on either side, and then we stack them onto our flexure design from the previous set of slides I was showing you. And what we can do then is trigger the muscle to contract. And this is the tendon, this is the tendon, this is the rigid skeleton, and that's the muscle piece. So this is the whole skeleton, tendon, muscle, tendon, skeleton assembly. And hopefully what you can see is that you're getting pretty, like, really good locomotion here. And the exciting thing about this, right, is that you essentially have the same strain output that you did from a tissue that was a whole ring, so 4 times this size. So by creating this sort of modular tendon muscle tendon actuator, we've enhanced the power to weight ratio of our actuators by over 4 times. And this sort of thing is very sensitive to a lot of different parameters, like how stiff is the tendon, how stiff is the muscle, how well clamped is the tendon to the skeleton, how much pretension is there. But we've done a whole range of optimization, and we have a bunch of data analysis in progress right now ahead of sharing this paper more broadly to basically describe how you can design these sort of actuators to get the most out of your muscle possible. So this is the kind of work we're doing in sort of thinking about how to take 3D muscles and convert them into useful functional work output. And then I'm going to pause. Okay. And then so now I'm going to shift gears and tell you a little bit about how we can also make planar soft robots using 2D muscle. But if anybody has any questions now on this 3D muscle part that I just showed, I'm happy to answer them now or later. So I'll give you a few seconds maybe to churn. Okay. All right. So if not, we can move on to the 2D category. So I started this talk by showing you a bunch of examples, right, of like thin film, like swimming robots that really inspired me as I was getting started in the field. And I started thinking about, like, why can't we make something like that with skeletal muscle? That's generally, we're thinking about skeletal muscle in like 3D chunks, like in our skeleton, like arms and legs and other things. But if we could build them in 2D, that would enable us to do cool things like twisting, coiling, swimming, folding, like other things you can do with a thin film type actuator. That would be very cool to do, right? But the big problem that we faced when we tried to do this is the fact that skeletal muscle, because it generates really large forces, it hates growing on a 2D surface, especially because most 2D surfaces that we grow cells on, like tissue culture plastic or glass, are very rigid. And cells, of course, are very, very soft. And so you can imagine if there's something that's very soft that's sitting on a very rigid surface, and then it's contracting a lot, the easiest thing for it to do is to peel off, right? And so that's what you see here. You're growing these muscle cells. So all these long lines are muscle cells, and then they're tearing away from their bottom surface because every time they're contracting, they're shearing or peeling off from that surface. So we thought for a long time, well, I guess you just can't grow them in 2D. They just don't like it, and they're always going to peel off. But over the pandemic, we ran out of a lot of ingredients to make our 3D muscles, and we had no choice but to try something else. And so what we did is we figured out that if you have a substrate that is stiff enough to support the muscles to differentiate, but soft enough to allow them to deform the surface without peeling off from their underlying layer, there is a sweet spot at which you can actually preserve skeletal muscles in 2D culture for a very long period of time. And so what we've done is essentially mixed fibrinogen and thrombin to polymers that help us form blood clots very quickly in our body. So fibrinogen is the monomer, and thrombin comes in and cleaves off little ends of the monomer so that they can attach to each other, and you can get this sort of cross-linked 3D hydrogel. And then by changing the amount of fibrinogen monomer you have in the matrix, you can significantly change the shear modulus and other mechanical properties of your gel. And if you do that across a wide range of concentrations, you can find one at which the muscle actually can survive. So here the muscle is shown in red. So each of these long lines is a muscle cell. You can find a modulus of fibrin, turns out to be around 0.1 kilopascal, so really, really, really soft, that the muscles can stick onto for, here we're showing up to 21 days, but we've had them in our group at least going to like 5 or 6 weeks at this point from both mouse and human cells. So now we can preserve muscle in 2D culture for a really long period of time. And then when the muscles contract, we can take a video of that contraction, computationally map the displacement over time, and extract how much forces the muscles are producing. And so what we see is that the muscles essentially produce almost the same amount of force as a 3D muscle tissue with much fewer cells. So we're maturing them much more efficiently. They're carrying around much less dead volume, and we're getting this really, really large actuation out of these bilayers of muscles sitting on a thin film of gel. But that seems nice, but in our 3D muscle tissues, all of the muscle fibers were pointing in the same direction because we were pulling them, right? And so they were aligning along the axis of tension. Here, that's not happening. And so you would get muscle fibers, some of them are pointing in this direction, some of them are pointing in that direction, and you don't really, have any control over that. So we're like, okay, in order to have control over that, what we actually need to do is to template the actuators via some sort of microtopographical cue. So we nanoprinted these stamps at the MIT Nano facility that allow us to essentially put really, really tiny lines or grooves into our gels that we make. And we can arbitrarily vary the geometry, right? So we tried a variety of different groove widths and depths and sizes, efficiently aligned both mouse and human muscle on these cells, on these grooves, and we saw that across a wide variety of groove sizes, ranging from 1x cell size, which is about 12 microns, to about 10x, about 125 microns, you can get these really nicely aligned, pink muscle fibers. But if you don't stamp or create any kind of topographical cues in your muscle, you can see that the muscle sort of grows anywhere and everywhere in whatever direction you want. And so if you have all of your muscle pointing in the same direction and then it contracts, then you get these really, really nice displacements that are correlated with the alignment of the fiber, versus if your muscle fibers are pointing in any different direction, you get these sort of really variable actuation patterns that are hard to follow and hard to control. And so this is just if you want everything pointing in the same line, but obviously if you're trying to make an actuator, you might want something a little bit more interesting or exciting. And so we started thinking, well, no one's really shown multi-degree of freedom locomotion from a muscle actuator before. And we started thinking about the fact that the iris muscles in our eyes kind of have a really cool multi-degree of freedom architecture, where you have a central layer of concentrically aligned muscle fibers. So here, each of these pink lines is a single cell. And so we have single fiber resolution patterning where we have a concentric layer of muscle and then an outer radial layer of muscle. And then when we shine light on this whole robot, we can get the pupil, right, to open or close based on how those muscles are contracting. So when the central concentric layer shrinks or contracts, you can close up the pupil of your eye. So this is a sort of principle we were trying to recreate. So we made this centimeter scale muscle-powered robot that looks like an iris. And then we showed that in different regions of the muscle robot, the radial zone, the interface zone, and then right around the pupil, you get very different actuation profiles that are matched to the alignment of fibers in that direction. And you can then shine light on the muscle and get the pupil to constrict. And the modeled version of how your bilayer contracts matches our our experimental values very well, because essentially it's just an active bilayer where your top layer is shrinking, and so then your bottom layer is just a passive gel. So you can model it really nicely, and you can get these really, really reproducible, nice motions. And right now we're operating with robots that are moving on the order of hundreds of microns, so still fairly small motion, and it's also fairly small tissue, so that's okay. But current ongoing work in my lab in this space is thinking about how to amplify this motion and get something that goes from the micron to the millimeter scale, so that we can start making swimming robots and other things that do more active and noticeable work that can be seen with the visible eye. So that work is still not published yet. This stuff is at least on bioRxiv, and hopefully it'll be out soon. But if you keep following this space, hopefully you can see more of what we're doing to amplify the motion of robots in 2D as well as 3D. All right, so I have about maybe 10 or 15 minutes before questions. So I'm going to show you a little bit of some work we're doing in the regenerative medicine domain, which is a little bit out of scope of robotics, but does apply very strongly to our robotics work. So the way my lab kind of pursues questions is that we're thinking big picture about what's missing from our models and what can we learn more about human biology. But sometimes the things we learn in our medical studies can apply back to our robotic studies, and likewise, things we learn in our robotic studies can feed towards our medical work. So let's look in that direction first. So when we had made all of these robots initially, we used them to understand some interesting things about biology. We said, okay, oh wait, I see something in the chat. Oh, great question. So hold on to this question, because that's what the talk is. So the question is about whether we see any forms of disease in the muscle, and that's exactly what I'm going to talk about right now. Okay, so when we have our muscle models in a petri dish, we can use them to understand more about biology, right? So we saw that if we exercised our muscles daily through daily light stimulation, we could make them get stronger. We have learned recently that our muscles do fatigue over time, but if we change their exercise training protocol, we can change their endurance behavior or response. And we've also learned that if if our muscle tissues get damaged, they can heal after that damage, and that exercising the newly regenerating tissue as it's recovering from that damage can accelerate the rate at which that healing happens. And so in our best sort of in vitro or petri dish type of models, we were able to get complete healing of our muscle actuators within about 2 days by shining light on them every day for about 30 minutes. And so we wanted to see if these things that we'd learned in our robotic systems could translate to models of injury and repair that you might see in a mouse. And so, or a human, but we can't study humans quite yet in a lab, so we do it in a mouse first. Okay. So one thing we've tried to do with our muscle actuators is, and my throat is starting to hurt, so I'm gonna, is think about if somebody had a traumatic injury, could we use the engineered muscle that we've developed in the lab to repair injury after trauma? And so to study this, what we do is we take a mouse, we induce a traumatic injury in that mouse's leg, and then we implant our engineered muscle as a graft into the site of that injury. And what we see here, so this is a cross section of the leg where each green circle represents a muscle cell, muscle fiber in cross section. This is the healthy leg of the mouse. So this is what it should look like. And this is what it looks like when we've put our engineered muscle in place. So you can see that they look very similar, right? You have these densely packed muscle fibers. The only thing that's different is that you have our muscle cells expressing an optogenetic ion channel that represents the fact that they're genetically engineered to respond to blue light, right? So these are the same cells that we use in our robots, but of course, normal animals don't have muscle that responds to light, so only our cells represent this, have this sort of tag. So now that we have this, that means that even after implanting the muscle inside an animal, we can still control it externally by shining light through the skin of the mouse. And so if we do this, we sort of directly activate the muscle graft, and we exercise or actuate it while it's implanted in the mouse and see if this can help recovery of the mouse over time. And what we saw is that mice that had these sort of actuated or exercised grafts essentially completely recovered the forces they were producing, and the mice were able to run at the same speeds they were running before within about 2 weeks of injury. But mice that were untreated were basically unable to match this running speed even after a month. So they remained slower. They had essentially a permanent injury, but mice that had these actuated grafts or were treated were recovered within about a couple weeks. And we asked ourselves, like, why might this be the case? The question somebody is asking here is about the intensity and frequency of the light. I think it's about 1 hertz, and the intensity I don't remember off the top of my head, but it's in the paper. But they are very, very no, I'm not even gonna guess it because I'm going to get the units wrong, but it's in the paper. But so we started asking ourselves, what's happening here, and why might actuating the muscle after it's being implanted be doing something to encourage recovery after injury? And I won't go into too much of the data of how we did this because it's maybe out of scope for this audience, but essentially we took up our muscle tissues, we digested them, and we ran them through a mass spec, and we looked at what proteins and signaling pathways are essentially being turned on or turned off in response to exercise. And what we saw is that when we exercise muscle tissues, we certainly make them more mature and more strong, but we also encourage cell signaling related to the growth of new blood vessels, which is called angiogenesis, and the growth of new nerves, which is called axonogenesis. And that this effect size could be modulated by changing the intensity of exercise. So when you have higher intensity exercise, you have more angiogenesis or more axonogenesis. And so what this means is that muscles could potentially, by exercising the muscles, you could be encouraging the growth of more nerves into that graft. And perhaps that is explaining why the mouse is now able to control the muscle with its own nerves and able to run at pre-injury speeds. So this is an interesting hypothesis, but very hard to prove in a mouse because you have a million other things that could be happening in that case. And so long story short, I won't go too much into this paper because it's a little bit more biological, but since that mouse paper, we have another paper that's coming out next week that shows that in a Petri dish model of growing muscles and neurons side by side in a Petri dish, if you exercise the muscles, you can actually definitively prove that the neurons, which are shown in green here, when they're not in the presence of exercise muscle, they grow a little bit. And when they're in the presence of exercise muscle, they grow a lot. And there's a lot of biology underlying, why this happens, what is the muscle doing during exercise to encourage the nerve growth and all these other things. But those are sort of biological questions, so I won't go too much into it. But the reason I wanted to share this story with you more broadly is to think about the fact that right now, all of our robotics work is just turning muscle on or off, right? Turning the whole muscle tissue on or off, or in the case of light stimulation, we stimulate some regions of tissue, but not the other. But in our bodies, we have like single cell control over muscle contraction. And we have this because neurons are touching every single one of those cells. And so we can get much, much precise, finer motor control if we would have neural integration. And then eventually, right, if you could connect those neurons to some sort of upper motor neurons from your motor cortex, you could start making more complex neural controlled machines that can make decisions about, go in this direction versus go in that direction. But to do that, first, you need to make sure that your muscle tissue can have nerves growing through it and connecting all of the fibers, and that you can actually actuate the muscle by controlling the nerves. And that has been a really, really hard challenge in the field. But the things that we've learned in this study, where we've learned that if we stimulate the muscle, we can actually encourage more nerves to grow out, grow towards, and innervate the muscle fibers, that's great. Now it tells us that we can use that sort of patterning technique to create more complex actuators that have neural control. So hopefully that made some amount of sense. I didn't want to spend too much time on this work for this audience, but just wanted to highlight some of the kind of back and forth crosstalk that happens in our lab all of the time between the things we study in the regenerative medicine context and the things we do in the robotics context. So quick recap before I take questions. The big picture goal, right, is to think about how we can build these sort of innervated muscle constructs in a lab setting. If we can there's some part of that that's more biological, like how do we get these cells to talk to each other? How do we get the nerves to grow into the muscle? How do we get the muscle to contract? And there's part of that that's really more robotics and mechanics. So how do I design the skeleton and the muscle tendon skeleton interface to ensure that I can get the most action and power out of my muscle and get some sort of useful, functional, and predictable output? So these are the kinds of things we're working on. And as Joel mentioned, we're fairly active on social media. So if you want to keep following these stories as they progress, I'm happy to keep sharing. And I'd like to wrap up by thanking all of the folks in the lab who have done all of the experiments that I've had the pleasure of showing you today. Thank you.
Thank you very much, Ritu, for the great talk. Let's open the stage for questions. You can either write in the chat and I can read the questions, or you can directly raise your hand and ask to Ritu. Let's start from the questions that were in the, well, I guess the first one about encountering malignant behavior was answered in this last piece.
A little bit, though. I think one part of it I'll add is that, here I just focused on muscle injury, but there's a lot of different types of malignancies that we could model using these sort of engineered tissues. So we also do work in my lab that I didn't show in this talk, thinking about how, for example, with different dystrophies, like muscular dystrophies, like genetic diseases, or with different neurodegenerative diseases like ALS, you can model those sort of diseases using these sort of tissues as well, and use that to understand how different types of therapies might restore muscle function or preserve muscle function for longer in patients that have these sort of conditions. We generally don't use those kind of cells for our robotics applications, right? Because you always want your healthiest and strongest muscle to be doing something exciting for you.
Yeah,
and then I see another
great
question here about how we integrate AI into our models and study. Short answer is we don't yet. I'm sure there's some interesting things we could do in terms of, we do generate a lot of very large data sets that can tell us a lot about muscle behavior in response to different types of training regimen. So it'd be really interesting if we could take the videos we generate and feed them into a model that can tell us, like, actually this type of exercise training or this type of composition of the media or food that you give to the muscle can maximize the performance of your tissue the most. So that's one example of how I could see it being useful. I'm sure there are others, but it's not something that I personally have the expertise to do. So I'm looking forward to opportunities to collaborate with folks on topics like this.
Yeah, actually, that connects to a question I also had. It's fascinating how you're able to propose interventions and see the effects of these interventions at various levels. But coming from control systems and decision making, usually that requires very precise models or some sort of system level viewpoint where you have the interconnection of all your components. And I was just curious, how do you go about that? Do you have somewhere let's say a logical interconnection of all of the various components in your system such that you can localize certain interventions? Or is it more, I'm aware that sometimes in biology, some things are more in kind of gut feeling, that it is like that, but one can still not prove why or how.
Yeah,
I'm just curious about the domain.
Yeah, no, it's a great question. It's very philosophical. I would say we're trying to get to that point. Right now, it's still very, I would say, gut feeling based, because I think the problem with the way a lot of tissue engineering is done right now, and what we're trying to move away from in the lab, is the fact that you sort of just like mix cells and proteins together, and then the only engineering that you're doing happens on day one that you make the tissue. And then after that, you're sort of at the mercy of stochasticity, right? And you're like, well, this one just turned out to be a great muscle and I don't know why, or this robot just turned out to be great and I don't know why. And I'm not saying we don't have that in my lab right now, we certainly do, but what we're trying to do is to understand how each variable, the cell concentration, the exact mode of exercise, the exact growth factor that you have in there, the interface that you have between the muscle and skeleton, we try to understand individually each of those components, like not only the muscle, but the muscle skeleton interface and the skeleton's mechanics and dynamics as well. So we're very much still, I mean, even this morning, I had a grad student, come to my office and be like, oh, this thing that we thought was going to move a lot didn't move very much at all. And so we're sitting there and I think, figured out what was wrong with our initial mathematical model and thinking of how we were thinking of this muscle flexure interface. So it's very much a work in progress. And I think you'll see our model sort of develop and change over time for sure. But we don't know yet, but we're in progress.
That's great. I mean, it's a very, very inspiring talk in this sense. I think also this, connecting back to the question about AI, this is probably where you can make use of all the data sets you collect. With some, I don't know about generative
what
AI,
it was about our star performers, right? That made them stars.
Exactly. There's another question from Subrata. Tissue engineering is a great work in your lab, as we learn from you. Thanks. I work on protein engineering, and it is interesting. Okay, it's not a question, it's kind of
Thank you.
That's great. Thank you, Subrata. Yeah, it's that's actually another question that I had. I know probably there's no answer, but did you find any parallelism with other disciplines that have totally different applications but similar issues?
Yeah, I mean, I think that one of the fields that is sort of parallel and interesting is the field of synthetic biology, where people are thinking about, how do I make some series of genetic modifications to a cell to get it to do some controlled behavior, right? And most of the time, people are thinking about using this to control the expression of certain genes. So as a therapeutic, you want to express more or less of a protein at some given period of time. But right now, we're engineering tissues. It would be great if they can give us cells that are also responsive to a variety of different things. So our initial building block has more functionality and modularity. So it's thinking at a different scale, but I think, we do it at a very small scale where, we make the muscle responsive to light, and that gave us a lot of capabilities. But I think that's just the very tip of the iceberg in what can be done to engineer the cells themselves.
That's actually a great idea. I know people here at Leeds are starting interacting with the Broad Institute on this topic. Maybe Munzer Dale, trying to bring some control theory perspective into the more interventions problems. But yeah, that's a super exciting field. I mean, I don't even work in that, but you convince me in this with this talk that this is worth pursuing. Very nice. Any other question for Ritu? I think you left you have here all your contacts in case people want to reach out. Thank you again very much for the talk. It was great having you here. And I will reach out once the recording is ready so that you can see it.
That sounds great. Thanks for all the questions.
All right. Take care and have a great day.
Thank you. Bye.
Bye-bye.